Hi, everyone. In today's session, we'll focus on an essential topic for anyone working in cell culture: how to maintain an aseptic environment. We'll break it down into three key areas: maintaining a sterile work area, handling materials in a sterile way, and following safe lab practices. So, why are these areas so important? Maintaining a sterile work area is the first crucial step in ensuring that your cell culture process is successful. Think of your workspace as the foundation of your experiment. If your work area is not properly disinfected and kept sterile, any work you do, no matter how careful, can easily lead to contamination. The key here is that contamination can come from any surface you're working on. It might be the bench, the laminar flow hood if not properly maintained, or even the equipment you use. Every time you work in a non-sterile area, you risk introducing harmful microbes into your cultures. These microbes can affect your cell growth and in some cases completely ruin your experiment. Therefore, regular cleaning with an appropriate disinfectant, like 70% ethanol, is essential. This doesn't just mean wiping down surfaces once in a while. This should be a routine part of your lab work. Additionally, minimizing movement and air disturbance helps prevent contamination from entering your workspace. Air turbulence can carry contaminants from the environment, so it's crucial to keep the workspace organized and undisturbed. Maintaining a sterile work area is about reducing the risk of contamination at the source. A clean workspace is the first line of defense in your cell culture process. Next, let's talk about sterile handling. Even if your work area is spotless, improper handling of materials, reagents, or equipment can still lead to contamination. When you handle materials, you're introducing variables that could potentially lead to contamination. For example, touching the inside of a container or using non-sterile tools can transfer microbes directly into your cultures. The importance of using sterile tools such as pipettes, pipette tips, and other lab instruments cannot be overstated. Even a small lapse in sterile technique can cause contamination and lead to unreliable or failed results. Additionally, we need to ensure that once our materials are opened, they stay sterile. This means handling them quickly and efficiently, and avoiding unnecessary exposure to air. Every second that a container is open or a tool is exposed increases the risk of contamination. Sterile handling is all about maintaining the purity of your reagents and tools, and it ensures that no unwanted microorganisms are introduced into your cell culture system. Finally, we need to talk about safe lab practices. These practices not only ensure that you're maintaining sterility, but also that you're working in a way that keeps both yourself and your colleagues safe. Safety in the lab goes hand in hand with sterility. Wearing the proper personal protective equipment is essential, not just for keeping your work sterile, but also for protecting yourself. This includes gloves, lab coats, and safety goggles. Changing gloves regularly, especially after touching potentially contaminated surfaces, is an important practice that prevents cross-contamination. Equally important is avoiding eating, drinking, or touching your face while in the lab. These are common ways to inadvertently introduce contamination to your sterile environment. It's also critical to handle materials quickly and efficiently to minimize their exposure to the open air. The less time they're exposed, the less risk there is of contamination. Safe lab practices are designed to maintain the integrity of your work and protect everyone involved. By adhering to these practices, you create a safer, more sterile working environment. To summarize, keeping your work area sterile, handling materials correctly, and practicing good lab safety are all essential for maintaining an aseptic environment. Contamination can come from many sources, so it's important to address all of these factors to ensure the success of your cell culture work. By following these guidelines, you reduce the risk of contamination and ensure that your work remains reliable. Thanks for watching. If you found this video helpful, don't forget to like, share, and subscribe. Feel free to leave any questions in the comments below, and stay tuned for more tips in our next video.